Hello everyone, SciencePod is a podcast that shines a spotlight on the latest scientific technology from Japan. Your host is Ren from Science Talk. Today, I'd like to talk about the computer that operates at the molecular level with individual molecules acting as computational units. This incredible future device is being realized by a research group at a Japanese university. When you hear the word computer, what comes to mind? Likely it's an electronic device like a laptop or a smartphone, something palm size or larger. However, what we're going to talk about today goes far beyond that conventional image. It's about an ultra-compact computer that operates at the molecular level. Why do we need such tiny computers? While conventional computers are incredibly powerful, they can't process information within our bodies at the cellular level. For the delicate tasks like early disease detection or delivering specific drugs only to the required location in our body, we need information processing at a much more microscopic scale. To tackle this challenge, DNA computing has garnered significant attention. DNA, as you know, is a blueprint of life, carrying our genetic information. But researchers have discovered that by leveraging DNA's unique properties, especially its sequence designability and specific complementary binding, it can be used for logic operation and computational processing, much like a programming language. Until now, traditional DNA computing methods involve mixing many DNA molecules in a test tube and utilizing their collective behavior for calculations. However, this approach made it difficult to observe the real-time movements of individual molecules, limiting precise information processing at the single molecule level. This is where nanopore technology comes in. This nanopore is a tiny hole in the membrane, and by capturing the electric signal generated when molecules pass through it, it allows for the highly sensitive, real-time observation of the individual molecules like DNA and proteins. It's a powerful tool that literally lets see molecular movements. The revolutionary device called sMOLU. This means Single Molecule Logic Unit, developed by Professor Ryuji Kawano's research group at the Tokyo University of Agriculture and Technology. This device was made possible by combining the nanopore technology and the DNA computing that I just introduced, resulting in one of the world's smallest molecule computing devices embedded in the lipid membrane. This sMOLU can perform a basic logic operation, an AND gate, at the molecular level, similar to what everyday computers do. An AND gate means that a particular result is achieved only when both conditions A and B are met. For example, imagine the security doors that only opens if both the correct key and the correct password are provided. So how does sMOLU achieve this AND gate functionality? And at the heart of this device is a Y-shaped three-stranded DNA structure, commonly called 3WJ DNA. This 3WJ DNA is engineered with recognition site for two different types of restriction enzymes, which are essentially molecular scissors that cut DNA at a specific sequence. You can think of it like a single rope marked with spots where only scissors A can cut, and other spots where only scissors B can cut. This Y-shaped DNA is securely fixed inside a tiny pore called alpha hemolysin nanopore. This nanopore is specifically suitable for DNA detection. Furthermore, the entire sMOLU system, including this nanopore, is integrated into the membrane of a giant unilamellar vehicle, which is an artificial cell-like a liposome made of a lipid bilayer, much like a natural cell membrane. And now for the AND gate operation. Two types of these molecular scissors. The restriction enzymes are introduced outside this like a liposome structure. The system is designed so that only when both of these restriction enzymes act simultaneously on the Y-shaped DNA fixed inside the nanopore, a specific DNA fragment is cleaved and released into the interior of this structure. If only one or neither of the enzymes act, this DNA fragment is not released. Once released, this DNA fragment then reacts with other DNA components prepared inside this structure, triggering a DNA amplification reaction. This amplification reaction is then visualized using fluorescent molecular levels. This allows the researchers to optically confirm with their own eyes whether the molecular level and operation was successfully performed, much like a light bulb turning on only when the correct conditions are met. So this system holds immense significance because it enables the direct observation and the control of the computational functions of a single DNA molecule using a nanopore technology. This truly represents a realization of the smallest unit of molecular computing. And while this research is still in its early stage, its potential is limitless, I think. The next steps aim to design even more complex molecular logic circuits or applying them to multi-stage computational processes. And this technology is expected to lead to evolutionary medical or diagnostic applications. This includes highly sensitive detection of disease markers by sensing environmental changes inside or outside cells, or even targeted drug release system that deliver precise amount of medicines to specific location in response to target molecules. The future where tiny molecular-driven computers protect our health is steadily approaching. That's all for today's Science Spot. This podcast is broadcast every on weekday morning in both Japanese and English. I hope today's discovery has changed how you view the microscopic world within us, even just a little. I'd like to listen to the podcast and post your notes and thoughts with the hashtag ScienceSpot. See you next time.